This is Chapter 27, the reproductive system. And the first thing we're going to talk about in this chapter is the primary sex organs that produce ova or sperm, and those are the testes in males and the ovaries in females. And these sex cells, either sperm or ova, are called gametes. These organs, the testes and the ovaries, they also secrete steroid sex hormones. So androgens are in males, primarily testosterone, and then you have estrogens and progesterone in females. Now, in both females and males, there are accessory reproductive organs. Those are the ducts, the glands, and the external genitalia that are involved with, in females, either producing the egg and development of the fertilized egg there, and in males, it's for production of sperm and conveying that sperm to the outside of the body. So we'll start with male reproduction, and we'll start with the testes. Now, the testes are surrounded by two tunics, and the tunic that is developed from the peritoneum is called the tunica vaginalis. And then under that is what's called the tunica albuginea, and that's more of a fibrous capsule that surrounds each testis. Now, testes are divided into lobes or lobules, and there's about 250 of these in each testis, and each one of these lobules contains 1 to 4 seminiferous tubules. Now, these seminiferous tubules are the site of sperm production, and each one's about 70 centimeters long, and surrounding these seminiferous tubules are cells that produce testosterone, and these are called interstitial cells. And these seminiferous tubules are composed of cells that are called sustentacular or Sertoli cells. We'll see this a little bit more detail in a minute. The blood supply is the testicular artery and vein, and the nerve supply is mixed, mostly sympathetic fibers from T10. Now, ejaculation is sympathetic, and erection is parasympathetic, and we'll talk about that later. So both systems are involved. Now, the scrotum is the pouch that contains the testes, and it is located in a position that's separated from the body because the temperature that's optimum for sperm development is about 2 degrees lower than body temperature. So that's why the testes tend to be located away from the core of the body. And there are 3 mechanisms that help to maintain this location and keep the temperature optimum for sperm production. So there's 2 sets of muscles. There's a muscle called the dartos muscle, which is a subcutaneous layer of smooth muscle that elevates the testes if the temperature decreases outside the body. So it'll move the testes closer to the surface of the body, or closer to the core of the body if the temperature surrounding the individual gets colder. The dartos, the cremaster muscle, contracts and draws the testes closer to keep the bodies warm. These 2 muscle groups work together to either move the testes away from the surface of the body or closer to the core of the body, depending upon the external temperature. The pampiniform plexus is a system or network of veins that helps to cool the blood before it reaches the testes. So with the 2 muscles and this network of blood vessels, the total function of this whole system is to maintain that temperature for optimum sperm development, which is 2 degrees below body temperature. Now, there's a system of ducts that carry sperm from the testes to the body exterior. So you start off with the sperm being produced in the testes themselves, in these sustentacular cells. Then they're conveyed from the testes by way of the efferent ductal, and that takes the sperm from the testes into something called the epididymis. And the epididymis is where they will stay and they will mature, and they are ejaculated from there by way of the ductus deferens. And this is a muscular tube that guides the sperm to the duct of the seminal vesicle, and then you go to the ejaculatory duct and then through 3 parts of the urethra. So those are all of the parts of the male duct system, and we'll talk about these a little bit more as we go on through the next few slides. There's the duct system in picture form, and I have taken yellow highlighter and mapped out the flow here of the sperm. It's produced here in this white structure here. This is the testes. Then you have that efferent ductal that's not really seen here, but that's the duct that takes the sperm from the testes and it goes to the epididymis, and the epididymis is where the sperm is stored. And then when the semen is ejaculated, you will have this ductus deferens, that the sperm goes all the way around here on top of the bladder, around and around, and eventually it will merge with a duct that comes from the seminal vesicle. Now the seminal vesicle, we're going to find out in a minute, produces secretion that adds more volume to the sperm. So you see the duct coming together down here with the vas deferens, and then it goes through this kind of walnut-shaped structure. This is the prostate gland. So when the seminal vesicle duct and the vas deferens come together, now it's called the ejaculatory duct as it goes through the prostate gland, and then it's going to combine with the urethra from the bladder, and then it becomes the prostate urethra, which is the beginning of the urethra. So the urethra in the male has 3 parts. The prostatic is first, that's in the prostate gland. Then it exits out from the prostate gland and fuses with a duct of another gland that's called the it's called the bulbourethral gland. So there's a bulbourethral gland. Those fuse together, and now the urethra, it changes names, and it becomes the spongy urethra. So the urethra actually has 3 names, and you're going to see a little bit of variation of the terms that are used. You have what's called the prostatic urethra, and sometimes this one here is the intermediate part of the urethra, may also be called the membranous urethra, and then you have what's called the spongy urethra, which you'll sometimes hear referred to as the penile urethra. So it's 3 parts of the urethra, but let's just back up again and go through this system one more time, this ductal system. Testes, efferent ductal into epididymis, vas deferens, then you have the ejaculatory duct, and then you have the prostatic urethra, intermediate part of the urethra, which is also called the membranous urethra, and the spongy or penile urethra. That takes care of that part. Okay, there are some disease processes that can occur with male reproductive structures. Testicular cancer is the most common cancer in males. It affects young males, primarily 15 to 35 years of age. Usually it's very painless. With most cancers, pain is not usually the first symptom that you see. I mean, it's not until later stages that you develop any type of pain. This term, cryptorchidism, refers to non-descent of the testes, and that's your primary risk factor for this type of cancer. It usually will display as a painless solid mass in the testes, can be easily palpated, and can be removed. Prognosis is very good with early detection and treatment, and 90% is cured by removal of the testes, and the term for that is called an orchiectomy. orchiectomy. The urethra conveys both urine and semen, but not at the same time. It has 3 different regions. This is what we were talking about earlier. So you have the prostatic urethra is surrounded by the prostate gland. The intermediate part of the urethra is also called the membranous urethra and the urogenital diaphragm. And then you have the spongy or penile urethra that runs through the penis and opens at the external urethral orifice. Now, we mentioned earlier about some glands that add volume to the sperm, and these are the 3 glands. Now, the seminal vesicle is the first one, and that actually produces about 60% of the fluid that will be added to the sperm as it's going through this ductal system. So it's a yellowish secretion that is mostly alkaline. It contains fructose, citrate, prostaglandins. A lot of these substances are for a food source for the sperm. The sperm cells can live up to 72 hours while they're traveling through the female reproductive tract, so they have a fairly long life and they need a food source. Citrate's the same thing. Prostaglandins, a lot of those are anti-inflammatory types of chemicals to prevent the female reproductive system from destroying them and deactivating them. Prosemenogelin is a coagulant, which causes the semen to coagulate, and then about 30 minutes after ejaculation, there's a serine protease that's produced from the prostate gland. That's your PSA, that's the sperm activator, which liquefies this kind of a gel-like substance and now enables the sperm to be released and be activated. So 60% of semen volume. Now the prostate gland adds a more thin milky secretion, makes another 30% of semen volume, and it contains more substances like citrate, enzymes, PSA, which is your sperm activator, or what they call serine protease, to liquefy the prosemenogelin. The bulbourethral glands, mostly mucus for lubrication, that contributes another 10% to the semen volume. So these 3 glands produce additional fluid to the sperm as they're released from the epididymis. Now, prostate disorders. You've probably heard about prostate cancer. It's the second most common cancer in men after lung cancer, affecting about 9% of men over the age of 50. And just like most cancers, it's asymptomatic in the early stages. But it can often metastasize to nearby lymph nodes, to the lungs and other organs. Digital exam screening, looking at PSA levels. PSA increases can indicate that there is a problem. So that's usually what they look for. Now, 80% of men with prostate cancer survive when it's detected and treated early, but if it spreads beyond the prostate gland itself and breaks through the capsule, then your survival rate becomes a lot lower in those cases. So getting it while it's in its early stages and still encapsulated is really critical. Now, sometimes treated with surgery, removal of the prostate gland, sometimes followed up with radiation, drugs. There's a lot of different ways to attack this depending upon the severity and the nature of where the cancer is located at the time when it is diagnosed. This picture looks a little convoluted, but if you print it, I don't think it'll be that bad. I think it's just because it's small here. But I wanted to just mention this part up here. If a couple's having problems conceiving, many times they will do a semen analysis in the clinical lab, and they'll take a look at the semen and see what the volume is. These are some of the things they're looking at. And usually the average ejaculation contains about 50 to 130 million sperm per ml. And the semen analysis is looking at the sperm count, how mobile the sperm are, what the shape of the sperm are, their morphology, the volume of semen, the pH, and the fructose content. If any of these things are altered or any of these things are off too far, it could mean that the sperm are not activated, they don't have enough food source, the pH is off, and that will affect their ability to be able to travel through the female reproductive tract. So these are things they look at in the semen analysis. The blue shows you the main pathway of sperm through the male ductal system, which we already talked about. But just to review again, here's the epididymis where sperm are stored. B is the ductus deferens, and then you go to the ejaculatory duct, and then you have the three parts of the male urethra, 1, 2, and 3 here. And then in green, I've outlined and kind of emphasized the 3 glands that add to sperm production, add to the volume. And 60% seminal vesicle, 30% prostate gland, 10% bulbourethral gland, and what they generally contain. Seminal vesicles, this says alkaline fluid containing fructose. Prostate gland contains PSA, that's your sperm activator, and PSA can also mark the course of prostate cancer. You look for elevations there to detect that type of cancer. And bulbourethral glands, basically mucus for lubrication. So that's an overview of everything that we've talked about to this point. Now, there is something you've probably seen commercials on from time to time called erectile dysfunction. And some of these medications that are out there, there's all kinds, Viagra, Levitra, Cialis, and there are many more. They kind of enhance the ability to create an erection. So here's what happens during the male sexual response. Sexual stimulation increases the production of nitric oxide. Now, nitric oxide causes the activation of guanylate cyclase. And guanylate cyclase is an enzyme that converts guanine triphosphate to cyclic guanine monophosphate. And cyclic guanine monophosphate is what causes vasodilation and causes the maintenance of an erection by allowing more blood flow into the penis. Now, there are phosphodiesterase inhibitors that the body produces that will break down this cyclic GMP and will cause these blood vessels to constrict, and then the person will lose the erection. So these medications are designed to block the effects of these phosphodiesterase inhibitors so that you can maintain the erection. So that's the idea, the biochemical process behind these medications that treat erectile dysfunction. So they're phosphodiesterase inhibitors. Now we're going to talk about the process of meiosis. Now, back in the beginning of AP1, you talked about something called mitosis. Mitosis was taking cells and dividing them into 2 identical daughter cells. Now, meiosis is a little bit different because when you produce gametes, you have to have the chromosome number reduced. We have 46 chromosomes in our cells. Those are our body cells. But egg and sperm have to be reduced to 23 so that when they come together and they unite and form a fertilized egg, now you're going to have 46 total. So you wind up with 23 from mom, 23 from dad, and the sperm and the egg come together, and those chromosomes that are homologous, that are matches, they come together, and they form the fertilized egg, which has the total number. Okay, so gamete formation involves meiosis. Now, I'm assuming that people have had biology, so you understand meiosis. And if you don't, and you want to enhance your understanding or go back and review, I would say to go back, find some YouTube videos that talk about the process of meiosis, and you can see this in more detail. I'm just assuming people have had biology, so this is something if you need to review, go back and do so. But it does differ from mitosis because instead of 1 cell division, you have 2, and only 1 round of DNA replication, which occurs in interphase. Most of the time when you're talking about mitosis, that's when you're talking about it happening. And because there's 2 consecutive cell divisions, instead of having 2 daughter cells, you have 4. And the function of meiosis is to take the number of chromosomes, 46, that you normally have, and reduce it to 23, so that when the sperm and the egg come together, you have a total of 46. Here it is in picture form, and this again is very small if you want to print it out and make it larger, but it compares mitosis over here with meiosis over here. And you can see with mitosis, you have your replication of your chromosomes, and then you go through your stages. You go through prophase, metaphase, anaphase, telophase, and you wind up with 2 daughter cells that are exactly alike. Over here in meiosis, you have your replication, and then you go through prophase, metaphase, anaphase, telophase, and you form 2 daughter cells. But something that will happen is you'll have a little bit more variation, because the pairs of chromosomes don't always go into the same cell. They kind of swap around a little bit, and there's something called crossing over, where the tips of the chromosomes will exchange pieces with each other, which makes more variation. And then you have another division, which is your meiosis 2, where these 2 cells, each one will divide into 2 more, and you wind up having half the chromosome number. That's called haploid, 23 chromosomes in each individual cell. So your differences are down here. And what you wind up with is, in mitosis, you have diploid cells. They have 2 copies of each chromosome. We have 23 different chromosomes, so you have 46 total. Meiosis produces haploid cells with half the chromosome number found in those diploid cells. And in mitosis, the daughter cells are identical to the parent cell, but in meiosis, the daughter cells are genetically unique. They say, the number I've always heard is the chances of producing an identical individual from gametes like this that are haploid is 1 in 72 trillion. That's your chance. So chances of having an identical individual produce, virtually impossible. In mitosis, there are 2 daughter cells formed, and in meiosis, there are 4 haploid cells produced. And meiosis consists of 2 consecutive cell divisions back to back in succession. Okay, so we're going to move on to talking about spermiogenesis. So we're going to talk about sperm production. We'll talk about egg production when we get to the female reproductive system. So sperm are also called spermatozoa, and the production of sperm occurs in the seminiferous tubules. Most body cells have 46 chromosomes, like we were just talking about. That's the diploid number, 2 of each individual one, and then 2 sets, 23 pairs of chromosomes, 1 from mother, 1 from father, and all the 23 chromosomes are different in the type of genes that they contain. So they all are individually unique, those 23 chromosomes. So they pair up. Homologous means that if it's chromosome number 5 from mom pairs up with chromosome number 5 from dad, and they have traits that are similar, and that's how they line up. Gametes have only 23 chromosomes, and that's the haploid chromosome number, only 1 member of the homologous pairs. So it's like a pair of shoes, you know. You have 2, the right and the left pair, and they go together. Well, if you separate them apart, you just have 1, and they match. They're a matching pair, but you only end up passing 1 of those off to the offspring. So you only have 1, and then it matches up. If it's mom, then dad's matches up with that pair. Spermatogenesis. Spermatogenic cells give rise to sperm. Now, this is where the immature spermatogonia are going to develop. They're in these sustentacular cells of the seminiferous tubules, and they're going to undergo mitosis and form 2 spermatocytes. Then those spermatocytes will undergo meiosis, and so the ones that undergo meiosis 1 are called spermatocytes. They form 2 cells, and those cells are are going to become primary spermatocytes, and then they will undergo the second meiosis, which will produce secondary spermatocytes, and then those cells will become spermatids, and then those spermatids are going to undergo the final transformation in the process of developing the mature sperm. So that's kind of an overview of the process, and we'll look at it in detail. So here you can see in this picture, this is a cell that's found lining the seminiferous tubules. And these are sustentacular cells, which here is shortened to sustentiocytes. Sustentiocytes is another term for sustentacular cells. Okay, so here's the basal lamina, and most of your immature spermatogonium are found at the margin here, at the lining, basement area of these sustentacular cells. Now, when they are activated, these immature spermatogonium, they start to migrate away from the bottom of this sustentacular cell. So they start to move closer to the middle. And they undergo mitosis, and they become primary spermatocytes. And that primary spermatocyte is going to undergo meiosis 1 and divide into 2. Now you see they're connected together by a bridge. And that's really important because they all stay together initially. And then they undergo meiosis 2, and these are the early spermatids. They're still connected together, and then eventually they will go through their final transformation. That cytoplasmic bridge will be disconnected. They'll be separated from each other, and then they go into the lumen of the seminiferous tubule, and they are conveyed from there into the epididymis. So there's the process of meiosis, and then the later stage of spermiogenesis. So I want you to remember that spermatogenesis is actually the process of meiosis. Okay, that's where you're reducing this chromosome number from 46 to 23. And then spermiogenesis is the next part. So spermiogenesis is the final transformation. Spermatogenesis is actually meiosis. So even though the words sound similar, don't confuse them. So here is spermiogenesis. So remember, you have 4 spermatids connected by these cytoplasmic bridges. So this is just looking at one of them. So it takes a while to produce a mature sperm. Actually, it takes about 74 days for a spermatogonium to become a mature spermatozoan. But a young man produces 300,000 sperm per minute, or 400 million per day. So there really is not any problem with numbers. So looking at spermiogenesis, you can see here's the nucleus that contains the 23 chromosomes. Now you see this little vesicle being formed at the top. It looks kind of greenish. This is called the acrosome. Now the acrosome is going to develop and contain hydrolytic enzymes that's going to be able to penetrate the female egg, get through the covering around the egg, and be able to gain access and latch onto the egg. So that's the acrosome. It contains hydrolytic enzymes. The mitochondria are going to aggregate behind the head of the sperm, and they will congregate here and form what's called the midpiece. Now the mitochondria are going to provide the ATP, the energy source, to be able to move this very long flagella. That's the tail. So when you get to the end, you have the acrosome at the head, the nucleus underneath that contains the DNA. You have the midpiece that contains the mitochondria, and a very, very long tail. That's spermiogenesis, and they will all be separated by the end of this process, and all the excess cytoplasm is reabsorbed back into the sustentacular cell. So there is a hormone connection that results in sperm formation. It all starts with the hypothalamus and the production of gonadotropin-releasing hormone that stimulates the anterior pituitary to produce FSH and LH. FSH, follicle stimulating hormone, goes to the seminiferous tubules, the sustentacular cells, and causes them to produce androgen-binding protein. Androgen-binding protein causes testosterone that's produced from the interstitial cells surrounding the seminiferous tubules. Testosterone will cause the immature spermatogonium to start to go through mitosis and go through their process of meiosis. So ABG causes the sequestering of testosterone. As the levels of FSH and LH, remember, causes the interstitial cells to produce testosterone. As those levels increase, inhibin causes the inhibition or the shutting off the production of anterior pituitary hormones. And so that's basically the process there, the hormone connection with the development of sperm. Now, testosterone is really important for secondary male sex characteristics too. And these are features that are induced in non-reproductive organs by male sex hormones, mainly testosterone. So the appearance of pubic, axillary hair, facial hair, enhanced growth of chest hair, deepening of the voice, skin thickening and becoming oily during adolescence. You see a lot of these changes occur rapidly. Bones grow, increase in density, skeletal muscles increase in size and mass, basal metabolic rate increases, and it's also the basis of sex drive or libido in males. Now we're going to move to the female reproductive system. Ovaries, these are paired organs on each side of the uterus that's held in place by several ligaments. The ovarian ligament is the ligament that anchors the ovary medially to the uterus. You have a series of other ligaments that support these internal reproductive structures. The uterine tubes, the uterus, and the vagina, make sure they're all supported in the abdominal cavity. The suspensory ligament anchors the ovary laterally to the pelvic wall. The mesovarium suspends the ovary in between, and actually the suspensory and mesovarium are all part of the broad ligament, which is the supporting ligament for all these internal female reproductive structures. There's a look at the female reproductive structures in picture form. The pear-shaped uterus, the dome shape at the top is called the fundus of the uterus. The terminal portion is the cervix. There's the vagina, uterine tubes. They expand as they go laterally and actually wrap around the ovary. However, be aware of the fact that there is no connection between the end of the uterine tube and the ovary. There's an open space. And so this means that any diseases that get through the female reproductive system can very easily go into the peritoneal cavity because there is that opening there. There's your broad ligament showing you the different parts of that ligament. There's your ovarian ligament right there, suspensory ligament that anchors the uterine tube to the lateral pelvic wall. Just a few of those structures there. The wall of the uterus, endometrium, is the innermost layer. The thick muscular wall is called the myometrium, and the outer layer is the perimetrium. There's like I said, this is the cervix. There's a canal that runs from the uterus down to the vagina, and that's called the os. This is the internal os, cervical canal, external os. Just some major structures there for you. And be aware, the female duct system have no contact with the ovary, so the oocyte is actually, oocyte's another term for the egg. It's cast into the peritoneal cavity. Some are lost there. This is your duct system. The uterine, or what they call fallopian tubes, are also called oviducts, the uterus, and the vagina. Here's the three layers of the uterine wall. Perimetrium is the serous layer. It's synonymous with the visceral peritoneum. Myometrium, interlacing muscles. This is your smooth muscle, which makes up most of the wall of the uterus. And then the endometrium is the mucosal lining. Now, because the oocyte is cast off from the ovary into the peritoneal cavity and hopefully whisked up by these Okay, so ectopic pregnancy is when you have an oocyte that becomes fertilized in the peritoneal cavity or distal uterine tube and develops there. Normally, these are aborted naturally with substantial bleeding. So that's an ectopic pregnancy. And then also, because of that disconnect between the ovary and the uterine tubes, pelvic inflammatory disease. Very easily, you can spread infection from the reproductive tract into the peritoneal cavity and may cause scarring there, which could lead to infertility. Common cancer, cervical cancer. 450,000 women worldwide each year are infected with this cancer, and it kills half. It's a very slow-growing cancer. Usually affects women between the ages of 30 and 50 years of age. Some of the common risks: frequent cervical infections, sexually transmitted infections, previously called STDs, sexually transmitted diseases. Now they call them STIs, and probably in a couple of years they'll change that too. Also multiple pregnancies. Pap smear is what's used for detection. And you want to really check with your physician, because I've seen a lot of variance on how many years in between you should have this, but generally speaking, every 2 years, 21 to 30 years of age. Every year, greater than 30, discontinue at 65 if negative for 10 years. But again, you might see some different schools of thought on how often and how long. Okay, the endometrium, that inner layer of the uterus, has 2 layers. The functionalis layer is the functional layer that is shed every month in women that are still ovulating and having menstrual periods. And this layer responds to changes in ovarian hormone cycles, and it's shed during menstruation. So there are 2 hormones in play here, remember, estrogen and progesterone, and they have functions at different parts of this 28-day cycle that we're going to talk about. The basal layer is a layer that stays. That's the innermost layer that stays behind, and it forms the new functionalis layer after menstruation, and it does not respond to the cycling of the ovarian hormones. This is the blood supply to the uterus. Uterine arteries that arise from internal iliacs. They're going to branch into arcuate arteries that run into the muscular layer of the uterine walls, the myometrium. And then they will branch into radial arteries in the endometrium that will go into straight arteries in the stratum bassalis and spiral arteries in the stratum functionalis. And these spiral arteries degenerate and regenerate. And these spasms cause the shedding of the functionalis layer during menstruation. So here's a look at the uterine wall. There is the uterine artery, and there's your arcuate artery, there's the radial artery, there's the smooth muscle of the uterine wall. Here are your straight arteries, and these are your spiral or coiled arteries. This is your functionalis layer up here of the endometrium. So the vagina or birth canal is a thin-walled tube that lies between the bladder and the rectum. It extends from the cervix to the exterior of the body. And the urethra is embedded in the anterior wall of the vagina. And this vagina provides a passageway for birth, menstrual flow, and it's an organ of copulation. So there's 3 coats or 3 layers. You have the adventitia, which is fibroelastic tissue. You have a smooth muscle muscularis and a stratified squamous mucosa. And the fornix is the upper end that surrounds the uterus. Now, external genitalia is called the vulva collectively. The mons pubis overlies the pubic symphysis. The labia majora is the hair-covered skin folds that are homologous to the male scrotum. Labia minora is the hair-free folds within the labia majora homologous to the ventral penis. And the clitoris is the erectile tissue, which is homologous to the penis. Mammary glands. These are modified sweat glands consisting of 15 to 25 lobes. They radiate around and open at the nipple. The areola are pigmented skin that surrounds the nipple, and suspensory ligaments attach to the breast and attach the breast to the underlying muscle fascia. Now, the lobes that are in the breast contain glandular alveoli that produce milk in lactating women. Compound alveolar ducts pass this milk to lactiferous ducts that open to the outside. Breast cancer is an invasive type of cancer. It's one of the most common malignancies that you will find in women. 15% of women will develop the condition, and 10% of cases are due to hereditary defects, including mutations to genes BRCA1 and BRCA2. 50 to 80% of people with these mutations will develop breast cancer, and also gives a greater risk of ovarian cancer as well. Breast cancer usually arises from epithelial cells of small ducts that eventually metastasize. Remember, epithelial tissue is rapidly dividing tissue, and most of your cancers come from this type of cell, this type of tissue type. Risk factors include early onset of menstruation or late menopause, no pregnancies or first pregnancy late in life, no periods or short periods or breastfeeding, family history of breast cancer. But one of the interesting things is, even with all these risk factors, 70% of women with breast cancer have none of these known risk factors. So it's a very difficult thing to predict. Early detection is very important by either self-examination, which is really important, and a lot of people don't do it, and also mammographies. Treatment depends on the characteristics of the lesion. Some of the treatments are radiation, chemotherapy, surgery, often followed up by radiation or chemotherapy to destroy stray cells if you have surgery. Drugs for estrogen-responsive cancers, tamoxifen, improves the outcome for premenopausal women with early or late stages for estrogen-dependent types of cancers. Now we'll go to oogenesis. Oogenesis is the process of egg formation. Now we talked about spermatogenesis, so now we're going over to oogenesis. And this is the production of female gametes, and it takes years to complete. It begins in the fetal period with the development of oogonia. These are your immature stem cells. They multiply by mitosis and they store nutrients. And the primary oocytes develop in the primordial follicles. Follicles are cells that surround the egg within the ovary. Now, primary oocytes begin meiosis, and they stall in prophase 1, and they sit there. And this is at birth. So at birth, you have these eggs that are just sitting there waiting to go through the rest of meiosis. This picture is going to take you through what's happening with the egg itself. This part talks about what's happening to the follicle that surrounds the egg, because that follicle is going to become very important in the female menstrual cycle and also if pregnancy should occur. So here's before birth. Your immature spermatogonium, your stem cell, undergoes mitosis, and it sits there waiting as a primary oocyte. And then in infancy and childhood, where the ovary is functionally inactive, the primary oocyte is arrested in prophase 1, which is present at birth, but is sitting there, not doing anything in the ovary. So when we think about this in numbers, because you know how much I love numbers, each female has about 2 million immature eggs in this state, sitting there at birth. And by the time that a female reaches adolescence, there's about 400,000 eggs that are ready to go. But during the reproductive life of a female, when she starts to have these menstrual cycles to the point that she ends up going into menopause, there's between 400 to 500 eggs that are ovulated. So there's way more than enough eggs that are there and available to go through the cycle and be ovulated. You have 400,000 that are ready, but actually only about 500 will actually go through the process of ovulation in the course of a female's reproductive life. So each month from puberty to menopause, this primary oocyte is going to, that's arrested in prophase 1, it'll complete meiosis 1, and then it'll be arrested in metaphase 2. Now you see that the excess DNA is cast off here. One interesting point I want you to remember, that each immature spermatogonium actually ends up producing 4 sperm cells, right? Each immature oogonium is only going to produce 1 egg. And the reason is because the only thing that's cast off when you go through meiosis 1 and meiosis 2 is the extra DNA. And that's just cast off and it's broken down within the ovary. You don't really have anything going on here that's going to produce more than 1 egg. We'll talk about some variations and some exceptions to the rule later. But this is meiosis 1, and you see that the DNA is cast off here, and there's the egg. Now, that secondary oocyte is going to be arrested in metaphase 2, and it stays that way. You go through ovulation where it's released from the ovary, and then it will not complete meiosis 2 unless fertilization occurs. And then it may or may not, this one may or may not actually divide. Doesn't matter because you're not using it anyway. But after you have fertilization by the sperm, then meiosis 2 is complete. And then the 2 nuclei will come together from the sperm and the egg, and you will have fertilization. So you wind up with 1 egg when you go through the process of meiosis in the female gamete. Now here's about the follicle now. The follicle surrounds the oocyte. So you have this primordial follicle, which is a couple layers of cells that protects the egg, and you can see it here a little bit larger. And then as you're going through meiosis over here, you can see that the primary follicle increases in the number of layers that it has, and then eventually it'll hollow out. It forms an antrum, which is a fluid-filled area in the middle. And then when ovulation occurs, this is the follicle that's left behind. There goes the oocyte that's going into the peritoneal cavity, eventually gets swept up into the uterine tube. But this follicle that's left behind is really important because for the next 10 days or so, ovulation occurs usually at mid-cycle around day 14. And for the next 10 days or so, this structure called the corpus luteum is going to produce hormones, estrogen and progesterone, to maintain the uterine lining for the next 10 days in case pregnancy occurs. Now, if the egg is fertilized within about 8 days, that fertilized egg and its covering, cells within that ball of cells is going to produce this hormone that's called human chorionic gonadotropin, or HCG. That HCG will maintain this corpus luteum for up to 3 months if pregnancy does occur. If there is no fertilized egg and there is no pregnancy, then after 10 days, the corpus luteum will die off because it's not being maintained by the production of HCG from the cells surrounding the developing embryo, and it'll eventually just die off, stop producing hormones, and becomes what's called a corpus albicans. And it'll leave a scar behind in the ovary. And when the hormone levels drop off, then that's what will produce the shedding of the functionalis layer and will cause the monthly period. So the corpus luteum is what keeps the lining maintained for at least another 10 days and producing hormones to complete the cycle. Okay, so the ovarian cycle, what's happening in the ovary? It's the monthly series of events associated with maturation of the egg. has 2 consecutive cycles or phases of this 28-day cycle. The first half is called the follicular phase. That's the period of follicle growth. That's days 1 through 14. And then ovulation, or ejecting the egg from the ovary, occurs in mid-cycle. And then the luteal phase, the period that the corpus luteum is active, that's days 14 through 28, maximum without fertilization. And as the levels drop off, then you have the menstrual cycle after that. So this picture is showing you the inside of the ovary and showing you the development of the egg, the primordial follicle. Those are your immature oogonium. And then you can see the follicle increasing in size. There's different aspects to the follicle. You have the theca folliculi, which produces the estrogen, progesterone. This is going to represent the parts that become the corpus luteum. The antrum is the open part that contains the fluid in between the egg and the follicle. And the zona pellucida is what surrounds the egg and protects it. So here you can see it's ready to be ovulated. There's the antrum. There's the zona pellucida around it, some other follicular cells. And you can see the follicle rupturing here, and it looks almost like an explosion, a volcanic explosion. Some women feel this. This little twinge of pain that occurs when the egg ovulates from the surface of the ovary is called Mittelschmerz. That's the term that was given to this twinge of pain that some women feel. And because of the scars left behind by multiple corpus albicans, that's the scar left behind from the corpus luteum, that what used to be a very smooth structure in a young female, by the time she reaches her late 30s to early 40s, the surface of the ovary is very pitted and scarred and almost looks like the surface of the moon. There's your corpus luteum. That's the follicle that's left behind after ovulation of the egg. There's ovulation right there in cartoon form. So just giving you a little bit more information on oogenesis and the development of the follicle. Here's the hormone connection in females. Same hormones involved, but a little bit different scenario here. We start off with gonadotropin-releasing hormone from the hypothalamus, and that's going to cause the production of estrogen and progesterone from the anterior pituitary gland. Now, these hormones are important with the development of the egg. Estrogen in particular, increasing levels of estrogen, once you reach a particular threshold or a particular point, that slightly elevated estrogen and rising inhibin from the ovary and elevated levels of FSH shut off the release of these hormones from the anterior pituitary. Remember that estrogen and progesterone come from the anterior pituitary. Doesn't mean that they're not being produced. They're just being sequestered here. Now, the dominant egg, that follicle, will continue to produce estrogen. And when estrogen reaches a second higher plateau, the next higher layer, then you will have ovulation, because that next higher level of estrogen is going to cause a surge of LH from the anterior pituitary gland, and that's what results in ovulation. And then the scar is left behind. It's called the corpus luteum. So here's the fluctuation of gonadotropin levels, and you can see here's FSH in red, LH in blue, and you can see the difference here around mid-cycle. This is around day 14 right here where the blue turns into green, and that surge of LH that's caused by the second elevated level of estrogen is what causes the surge that results in ovulation. And remember, ovulation is the release of the egg. Here's what's going on with the ovarian cycle. So this is the first half in blue, second half is in green, and the first half is development of the egg in the follicle, and you can see at around day 14 ovulation occurs. So this is the follicular phase where the follicle is being developed, and then your second half, the second half of the ovarian cycle, is called the luteal phase, and that's when the corpus luteum is functioning and producing hormones to maintain the uterine lining in case fertilization and pregnancy occurs. Now this is the uterine cycle, which is different from the ovarian cycle. The ovarian cycle is what's going on with the ovary. This is what's going on with the uterine lining. This is the cyclic changes in the endometrium in the response to fluctuating ovarian hormone levels, and there's 3 phases. Day 1 through 5 is the menstrual phase, where you're shedding that innermost lining, the functionalis layer of the endometrium. Days 6 through 14 is the proliferative stage. That's where you're developing a new functionalis layer. And days 15 through 28 is the secretory phase, post-ovulatory phase. That is a constant length of 14 days. So that is where you're secreting hormones from the corpus luteum to maintain this layer in case pregnancy should occur. This is the cycle that's going on within the uterus itself. Here's what's going on with the fluctuation of ovarian hormones. First half, second half of this cycle. So estrogen has its greatest influence in the first half of the monthly cycle. You know that the surge of estrogen at mid-cycle causes the burst of LH, which results in ovulation. Progesterone is pretty flat here. Progesterone really has its effect in the second half of the monthly cycle, because what it does is it prepares the uterus for implantation and receiving a fertilized egg. So this one is in preparation, preparing the uterus to be maintained in case pregnancy should occur. So estrogen is primarily first half, progesterone second half. This is what's going on inside the uterus, inside that lining, and this is the changes that occur in that functionalis layer. So the menstrual phase is when the layer is shed, and then you have the proliferative phase, building up a new layer, and then preparing for implantation. So menstrual functionalis layer is shed, proliferative, building the new one, begins at this secretory layer begins after ovulation. Enrichment of the blood supply and glandular secretion of nutrients prepare the endometrium to perceive an embryo. Both the menstrual and proliferative phases occur before ovulation, and together they correspond to the follicular phase of the ovarian cycle. The secretory phase corresponds in time to the luteal phase of the ovarian cycle. So you can very easily superimpose these two cycles, the uterine and the ovarian, time-wise with each other. So they're very similar in their time frames. So menopause is what happens when estrogen levels decline as people age, usually somewhere around the 40s. Atrophy of reproductive organs and breast occur. Irritability and depression in some. Hot flashes as skin, blood vessels undergo intense vasodilation, gradual thinning of skin and bone loss, and increased total blood cholesterol levels and falling HDL. So that is the end of this chapter.